are cells in our genes inside the cell, and as the antibiotic is inserted into the cell, the antibiotic kills the anti-resistant, or the anti-resistant genes stay as the cells that aren't resistant to the antibiotic, they die, and then the anti-resistant genes, they duplicate and form in a new cell, and then those genes can either be transformed to another cell or they can just duplicate. Anti-resistant bacteria is E. coli, and that's found in some hospitals. Doctors are trying to treat it with Colston, and when they treat it with Colston, the E. coli is becoming anti-resistant to it because of its MCR1 gene, and it's causing it to not die when it's treated with Colston. Another example is the MRSA infection. Doctors have been trying to treat it with penicillin, and it's been found recently that over 80% of the drugs that they've been using to try to treat it have been not effective.
One example of this is fruit flies. Recently, they have become resistant to the pesticides that are being used against them, especially malathion, which is used for fruit flies that are in orange groves. Another example of this is the Colorado potato beetle. The beetle has become resistant to 52 different types of insecticides that have been used against it and has caused havoc on the Colorado potato.
For our project, we chose 2 different locations throughout the school to test the effectiveness of 2 different antibiotics. The 2 locations we chose were the door and the water fountain because we thought those get used quite a bit and we'd like to see how the effects of these antibiotics would have. Now we were just going to show you the process we used to test our results. So first was our controlled, where you just get the Q-tip a little wet and you rub it down on the door, and then you put it in your Petri dish, and we put it in the incubator and we saw the different things. Our next one was just the Clorox wipes. We took a little bit of the Clorox wipe, wiped it down, did the same thing, got the Q-tip a little wet, wiped it down, put it in the Petri dish, and let it incubate for a couple days and see. Lastly, we used rubbing alcohol. So on one side of the Q-tip, we'd get it wet with the rubbing alcohol, rub that spot down, then we'd flip to the other side, get that wet with just water. Put the same spot down, put it in the Petri dish, and let it see. And then we did these exact same steps on the water fountain, and you will see our results here on the next.
On the left, we have our controlled for the door, and then we have after we used our Clorox wipe and then swabbed it, and then we have after we used rubbing alcohol and swabbed it. And it looks like for the door, when we used Clorox wipes, the green mold that was seen on the controlled looks like it increases, and the pink yeast looks like it decreases, although they're a lot more spread out. And then for the rubbing alcohol, it looks like the green mold increased by a little bit. There's 2 dots here. There's only 1 in the controlled, but the pink yeast seem to clump up together more and not be as abundant as the controlled. And then you see little dots of round yeast on the rubbing alcohol, which you don't see on the Clorox. So it can be assumed that the rubbing alcohol is worse against the yeast than the Clorox wipes are.
So on the right here is then our water fountain, and as you see, the controlled was actually a lot more dirtier than even the door was. It had that pink yeast, and then it had a little bit of black mold here in the bottom left. Then the Clorox wipe looks like it did a pretty good job. It rubbed out basically all the black mold. It had a little bit of round yeast in here, and then the pink yeast was cut down, but there also is still some there. It doesn't have as defined shape as in the controlled. And then the rubbing alcohol looks like it did the best in our whole experiment on the water fountain. It does have a little bit of peak yeast, but it doesn't have any of the mold, and it just looks a lot more cleaner than those 2.